Dear students, we shall see about the fermented vegetable products. The lactic acid fermentation of vegetables, applied as a preservation method for the production of finished and semi-finished products, is considered as an important technology because of its capability to improve the nutritive value of the product, palatability of the product, acceptability of the food, microbial quality, shelf life of the fermented product. Vegetables are chiefly fermented so that they can be used in seasons when they are not available. Fermentation of vegetables are done in regions of India where the vegetables are not available throughout the season. When a particular vegetable is not available throughout the season, they are fermented and they are used in the off season. When the vegetables are fermented, there is improvement in the keeping quality, in the nutritive value, and also the palatability and acceptability of the product. Moreover, this is a remarkable procedure to store the perishable vegetable in the absence of cold storage or refrigeration. Vegetables are a perishable product. When the vegetables are not stored in an ambient condition, they decay or the quality of the vegetable is deteriorated. In order to prevent the deterioration of the vegetables, they can be fermented. Majority of the rural people cannot afford to canning or to frozen foods. There is no availability of cold storage, and therefore the vegetables were fermented to be used after a period of time. Fermented vegetable-based foods have a number of medically beneficial properties. Apart from having improved shelf life, the fermented vegetables also had medical benefits. Fermented products are consumed as a traditional food by the ethnic people of northeastern states of India. So this is considered as one of the traditional and the most ethnic food, and it is chiefly consumed in large scale by the people living in the northeastern states of India. The preserved fermented vegetables are consumed in the long monsoon season, during which the fresh leafy vegetables are not available in plenty in the mountain regions. In India, bamboo shoots are harvested annually in Sikkim, 26.2 tons, Meghalaya, 435 tons, and Mizoram, 426.8 tons. Bamboo shoots are one of the popularly fermented vegetable in the northeastern states. Bamboo shoots are low in fat and cholesterol, but they are very rich in potassium, carbohydrates, and fiber. Many nutritious and active materials such as the vitamins, amino acids, and antioxidants such as flavones, phenols, and steroids can be extracted from the bamboo shoots. Common ethnic bamboo shoot fermented products are mesu, soydan, soybum, soyjim, yakung, yap, hiring, and lung sage. Bamboo shoots are considered as a nutraceutical medicine because they have low fat, they are high in fiber and vitamins such as vitamin C and vitamin E. In the Ayurvedic system, bamboo shoot is recommended for patients having piles and burning sensation during urination, and is advised to take the shoot churna, which is the powder, along with honey. Northeast Indian people use the fermented bamboo shoot products such as mesu and soybum. The dish is well suited and are consumed along with rice and chapatis. The lactic acid bacilli is the dominant microorganism in the ethnic fermented vegetables and bamboo shoot products. Pediococcus pentasaceus, Lactobacillus celubiosus, Lactobacillus plantarum, Lactobacillus fermentum, Lactobacillus brevis, Lactobacillus mesenteroides, Lactobacillus lactis, Enterococcus fecum, and Penicillium acidilacti or predominant lactic acid bacilli found in the fermented vegetables. During the fermentation of the radish taproot product called a sinki, Lactobacillus plantarum utilizes mannitol to remove the bitter flavor from the finished product. Bamboo shoot based fermented foods contain Lactobacillus plantarum, Lactobacillus brevi, Lactobacillus corniformis, Lactobacillus delbrueckii, Leuconostoc fallax, Lactobacillus lactis, Lactobacillus mesenteroides, Enterococcus durans, Streptococcus lactis, Lactobacillus casei, Tetragenococcus halophilus, and Lactobacillus fermentum as predominant lactic acid bacilli species. They also showed functional probiotic properties. Probiotics are those that contain the live microorganism in the food. During fermentation of the bamboo shoot, Lactobacillus lactis help in reducing the cyanogen glycoside, which is a toxic component that causes major diseases of the nervous system, goiter, and miscarriage. Let us see the fermented vegetable products. The first one is called as the mesu. Mesu is a traditional fermented bamboo shoot pickle with a sour acidic taste of Darjeeling Hills and Sikkim. Locally grown young edible shoots, choya bands, they are called as the choya bands, the karati bands, and the balu bands. They are all the different varieties of bamboo. They are defoliated, the leaves are removed, they are chopped finely, they are pressed tightly into a green hollow bamboo stem. When the bamboo stem is cut, they are hollow in the center portion. So the young and the edible shoots, they are cut and they are packed in the hollow portion of the bamboo stem. The tip of the vessel is covered tightly with leaves of the bamboo or some other wild plants, and they are allowed to ferment under natural anaerobic condition for a period of 7 to 15 days. Completion of fermentation is indicated by the typical mesu flavor and taste. The dominant producer of mesu is the Limbo women of the ethnic Nepal community. Mesu is eaten as a pickle. Mesu pickle is mixed with edible oil, chilies and salt like we do, and is kept in a closed jar for several months without refrigeration. So the keeping quality has improved because of the fermentation process. Mesu, kept in a green bamboo vessel, loosely capped by leaves of fig plant, tried by straw, is commonly sold during the rainy season in the local markets of Darjeeling Hills and Sikkim by the Limbo women. Mesu contains the following microorganisms: Lactobacillus plantarum, Lactobacillus brevis, Lactobacillus curvatus, Leuconostoc citrum, Pediococcus pentosaceus have been found in the samples of mesu. This chart shows the mesu preparation. Young bamboo shoots are cut. They are the outer inedible casings are peeled off. They are removed, the outer portion is peeled off. The inner part is chopped, washed with water and cut into pieces. They are pressed tightly into the fresh bamboo vessel. Here the fresh bamboo vessel means the stem of the bamboo, which is hollow, and it is made airtight by capping the mouth of the bamboo vessel. This container is kept in an upside down position, and it is fermented for about 7 to 12 days. After this period, the mesu is obtained. The next product is called as the soydan. Soydan is a fermented tip of the matured bamboo shoot, which is popularly consumed in Manipur. Tips of the matured bamboo shoot are collected. The outer casings and the lower portion are removed. The whole tips are submerged in water in an earthen pot. They are cut and they are kept in an earthen pot where there is some water. The liquid soyjim, that is the starter culture from the previous batch, is taken. It is diluted in the portion of 1:1. It is covered. It is added to this earthen pot. The earthen pot is covered and fermented for 3 to 7 days at room temperature. Leaves of Garcinia pedunculata, which is a locally available herb, may be added to this bamboo shoots, which is fermenting in the earthen pot to enhance the flavor of soydan. After 3 to 7 days of fermentation, soydan is removed. Soydan can be kept in closed container at room temperature for about a year. The dominant producer is the Métis women who are supported by their men. The microorganisms present in soydan are Leuconostoc phallax, Lactobacillus brevis, Lactobacillus lactis, Leuconostoc citrium have been found in the samples. This chart shows the preparation of soydan. Tips of matured bamboo shoot are taken. The outer inedible casings are peeled off. The inner portion is cut into small pieces. The unwanted lower portion is removed, washed with water. They are put in an airtight container or an earthen pot, and a small amount of starter culture is added, which is the sour liquid called as the soyjim of the previous batch. To this is added leaves of Garcinia pedunculata, and they are fermented for about 3 to 7 days. After 7 days, they are removed from water-filled container, and this product that is obtained is called as the soydan. The next product is called as the sinki. Sinki is an acid, non-salted, fermented radish taproot consumed as the base of soup or as a pickle by indigenous people of Nepal, Darjeeling, Sikkim, and Northeast India. For consumption, sinki is immersed in water for 2 minutes. Sinki is the fermented product. The fermented product is immersed in water or soaked in water for 2 minutes, and after it is removed from water, it is fried with salt, onion, and green chili. The fried mixture is boiled in rice water and it is consumed as a soup along with the main meal. Sinki contains 14.5% of protein, 2.5% of fat, and 11.3% of ash on a dry basis. This fermentation is initiated by Lactobacillus fermentum, Lactobacillus brevis, followed by Lactobacillus plantarum. Sinki is prepared by pit fermentation method. This is very interesting method of fermenting. About 1 meter pit of same diameter is dug in a dry place. So they make a pit in the ground. The pit is cleaned and it is plastered on the inside with mud and it is warmed by burning. After removing the ashes, the pit is lined with bamboo sheath and paddy straw. So there is a pit made in the ground and it is plastered with clay and the clay is burned. And after this, it is lined with the bamboo sheath or the paddy straw. Radish taproots are wilted for 2 to 3 days. They are left in the sun for about 2 to 3 days, and they are allowed to wilt. They are crushed, dipped in lukewarm water. Lukewarm water is water which is at about 60 degrees centigrade. They are squeezed and they are pressed tightly into the pit. So after the radish is wilted, it is crushed, it is dipped in water and it is squeezed. Then it is pressed into the pit. Then it is covered with dry leaves and weights are applied on the top by heavy planks or stones. The microorganisms present in Sinki were Lactobacillus plantarum, Lactobacillus brevis, Lactobacillus casei, Leuconostoc phalanx. Sinki preparation: radish taproot are taken, they are wilted, crushed, pressed into the pit, they are covered and they are weighted down. They are allowed to ferment for about 22 to 30 days, and after this they are cut into pieces and they are allowed to dry in the sun for 3 to 5 days. The product thus obtained is called as the Sinki. The next product is called as the sauerkraut. Sauerkraut is a type of preserved fermented cabbage. The Romans, way back in the ancient period, they knew about sauerkraut. Columbus, the very popular traveler, nourished his sailors with sauerkraut to make them resistant against scurvy and other diseases, because cabbage is a rich source of vitamin C, and Columbus undertook long voyages in order to prevent scurvy, which is vitamin C deficiency. He fed his sailors with sauerkraut. The Greek medic Hippocrates recommended sauerkraut against overweight, metabolic disease, and for smooth detoxification, that is, removal of the toxins accumulated in the body. With the migration of people, this food came from China to Europe. This chart shows the preparation of sauerkraut. Fresh cabbage is taken, they are cleaned, washed, graded, they are cut or shred, salt is added, and fermentation is allowed to take place with the help of the natural microflora, during which process lactic acid is produced. They are then packaged in glass jars and stored at room temperature. Shredded cabbage is placed in a jar and salt is added. Mechanical pressure is applied to the cabbage. So at the top, we place some weights. Mechanical pressure is applied to expel the juice, which contains fermentable sugar and other nutrients suitable for microbial activity. The first microorganism to start acting are the gas-producing cocci, Lactobacillus mesenteroids. These microbes produce the acids. When the acidity reaches about 0.25 to 3%, the bacteria's growth slows down and they begin to die off. Because it becomes more acidic, the environment is not conducive for the growth of the microorganism, although their enzymes continue to function. The activity initiated by the Lactobacillus mesenteroids is continued by the lactobacilli, Lactobacillus plantarum and Lactobacillus cucumeris, until an acidic level of 1.5 to 2% is attained. Thus, high salt concentration and low temperature inhibit these bacteria to some extent. Finally, Lactobacillus pentoaceticus continues the fermentation, bringing the acidity to 2 to 2.5%, thus completing the fermentation. The end products of a normal sauerkraut fermentation are lactic acid along with small amount of acetic and propionic acid. A mixture of gases like carbon dioxide, which is a principal gas, small amount of alcohol, and a mixture of aromatic esters are present. The acids in combination with alcohol form esters, which contribute to the characteristic flavor of sauerkraut, which is the preserved cabbage, or we call it as in the common name as cabbage pickle. The acidity helps to control the growth of spoilage and putrefactive organisms and contributes to the extended shelf life of the product. Because of the acidic nature, the shelf life is extended. Changes in the sequence of desirable bacteria or the presence of undesirable bacteria alter the taste and quality of the product. The optimum temperature for sauerkraut fermentation is around 21 degrees centigrade. Salt plays an important role in initiating the sauerkraut process and affects the quality of the final product. So the amount of salt added will affect the quality of the final fermented product. The addition of too much salt may inhibit the desirable bacteria, although it may contribute to the firmness of the kraut. If there is too much of salt addition, the microorganism growth will be inhibited, but the product obtained will be firm. The principal function of salt is to withdraw juice from the cabbage, thus making it more thus providing a more favorable environment for the development of the desired bacteria. Generally, salt is added to a final concentration of about 2 to 2.5%. In order to produce sauerkraut of consistent quality, starter has been recommended. Not only do starter cultures ensure consistency between batches, batches are when they are prepared in large scale. To get the consistent quality, starter cultures can be used. They speed up the fermentation process as there is no time lag while the relevant microflora colonize the sample. It is possible to use the juice from the previous sauerkraut fermentation as a starter culture for subsequent fermentation. So in any fermented product, the leftover of the earlier product or the earlier batch can be used as a starter culture for the next batch. Often the use of old juice produces sauerkraut which has a softer texture than the normal. So when such starter cultures are used, the product obtained is having a very soft texture. Thank you. In this session, we saw about the various fermented vegetable products that are commonly consumed in India. Thank you.